Hey guys, perhaps something different today. I thought I'd talk about vaccines. I'm an immunologist, so I think there may be some interest into understanding vaccine a bit more. There's still a little bit of talk about COVID vaccines and what happened a few years ago, and and sometimes I feel that a lot of people miss the basics on vaccines. So I thought I give a little primer on vaccines, and then we'll see where we go with that. If there's a lot of interest, we can perhaps dig it a bit deeper. Immunology is quite a complex. It's a very complex field of biology, um, and as immunology progressed in the last decade or so, it's also been immunology kind of grabbed like the entire field of physiology. I feel you would ask anybody on the street, what is immunology, what's the immune system? Well, it's to fight off diseases. Perhaps they're aware of its ability to fight cancer, but then that's that. But the problem is now almost like virtually any cell in the body is contributing to immune function. So there's a constant interaction between, well, what you would consider professional immune cells and any other cell. So everything signals, everything crosstalks. So what we consider to be immunity, it amounts to the entire physiological system of the body almost. So the field of immunology has grown a lot, and more recently, there's also like the entire nervous system interacts with the immune system as well. So as immunologists, like suddenly we were faced with understanding the entire organism, or sometimes, of course, you need to focus on a particular subject so that you can actually go into depth. You cannot cover it all. But the field itself has grown tremendously. And so vaccines is obviously something that people are interested in because they're facing it's something that is of interest because they get vaccines, or they are thinking about getting vaccines, or they may not fully understand what a vaccine is. So I thought a little primer would probably be good. And feel free to leave comments if there's anything that you would like me to cover a bit more in detail. So let's just start with let's just put one thing straight. So vaccine, it doesn't have a really a tight definition. Um, so when I hear, well, the COVID vaccine, yeah, well, vaccine is supposed to protect everyone or shield everyone. Well, that's not part of the definition of vaccine. So ideally, if you roll out the vaccine to millions or billions of people, ideally, of course, you would assume that it's so good that it is highly protective and all of that. But it's not intrinsically a definition of a vaccine. A vaccine is not supposed to do anything. Well, it's designed to do something, but at the end of the day, what it actually does or how good it is, a bad vaccine is still considered a vaccine. So the term itself does not incorporate any measure of efficacy whatsoever. Um, and this is because the definition of a vaccine. So typically, what comes to your mind probably is a prophylactic vaccine. So something that you take and protects you from contracting a disease, for instance. But there's also therapeutic vaccines. So vaccines where you already have a disease, like cancer vaccines. So I was still part of a cancer vaccine study where people already have cancer, and then you try to trigger the immune system to fight off what's already there, and that's also considered a vaccine. So you have prophylactic, you have therapeutic. So that by itself tells you, and most of the cancer vaccines, the therapeutic ones, like they're not particularly good if they're not combined with anything that really enhances their efficacy. So they've been bad for a while. So you already have that distinction. And then, so where it becomes complicated is the vaccine intrinsically is designed to help you with something. So either it's there to help you with something that might come. So it's a prophylactic vaccine against an infection that you might contract, or it's designed to help you fight off something that's already there, a therapeutic vaccine against cancer, for instance. So what the vaccine does, every vaccine is going to be very, very individual in what it does. And that's where you go a little bit into immunology, because there are vaccines that are more or less designed around a T cell response, and then there are vaccines that are designed around a B cell response. So these are two key cells of the immune system, of the adaptive immune system. So the part of the immune system that has memory, although the innate immune system also has memory, but it's kind of different. So I'm not going to go into it's called trained immunity, but it has different mechanisms. So let's just talk about the adaptive immune system. So most of you will be familiar with antibodies, which are produced by B cells. So if you know that to prevent something, or where usually the B cell vaccines are prophylactic, so to prevent something you just need to have a lot of antibodies in your circulation, then you're going to target your vaccine or design it around a B cell response. Mind you, T cells will always be involved as well. Same thing with if you design a T cell vaccines, there will be some B cell responses as well. But in the design, you clearly need to understand what is protective. And that's an interesting one, because you have to understand, what do I want to achieve specifically? You don't want to just activate the immune system, or just like, oh yeah, just throw something in the organism and just, oh yeah, she'll be right. No, you need to fully understand mechanistically what is going to protect that person. And that's not necessarily trivial. Just to give you an example, so I worked in the field of HIV, more related to transplantation, so not really related to vaccines, but I was aware about the vaccine work going on, like that was roughly 15 years ago, where the entire field was clearly hunting for T cell vaccines. So T cells, part of them are cytotoxic, so we call them CD8 T cells. They express a marker that's called CD8, and they're cytotoxic, so they're going to kill virally infected cells. That's what a T cell does. Because at the time, it was believed that because HIV mutates one of its outer proteins so rapidly, that having an antibody response was also based on the structure of the outer protein. It was believed that an antibody that would bind what doesn't mutate all the time, it wouldn't be able to bind there. So that's why all of the eggs, or most of the eggs, were basically put in a T cell basket, and people designed a T cell vaccine. And then suddenly the field swapped, because there were some of these people that had contracted HIV that were controlling the virus in a weird way, and then then scientists were analyzing, or were trying to understand, how is that person able to control the virus? We've never seen that, So there were a few around, and those people somehow were able to have a B cell response, to produce antibodies that were able to do something that scientists believed was not possible. And so it's almost that the scientists try to retro design. Okay, that person is able to do that without any vaccine, without anything. They had the infection and then they were able to do that. How can we retro-engineer that thing, the biology, and figure out how to induce that in other people? So the field switched from a T-cell based design to a B-cell based design, with the goal in mind to produce what some people on Earth are able to do by themselves. So it is critical to any kind of vaccine design to kind of know what can induce protection, because the immune system is extremely complex. If you don't know what specifically you want to induce, or if you don't know specifically what is protective, you're going to fail, because you might induce an immune response, but you don't have protection. And so for COVID, for instance, like people were, as I understand it, designing the vaccines around B-cell responses, around antibodies, which makes sense for most infectious diseases. I heard a few podcasts where people were saying, well, I was hunting for a T-cell vaccine, but somehow it wasn't selected. So I can't remember which podcast it was, but it was actually quite interesting that there might have been some decision making on a very, very high level to say, no, we don't want that T-cell stuff. We go straight for B-cells. A T-cell wouldn't have been bad. So the difference between the two in an infectious disease scenario is that the antibody is floating around in your blood. So if the virus is floating around in your blood, the antibody is gonna bind the virus, hopefully neutralize it to some way. Not all antibodies will be protective, right? So you need an antibody that neutralizes or incapacitates the virus so that it cannot enter your cell. So for instance, the monoclonal antibodies that were on the market were designed to do that. So they were binding the virus in such a way that it was unable to replicate or to infect the cell and then replicate. And then the antibody has other functions. So on the front end, it binds whatever it's supposed to bind, and on the back end, it can interact with cells. So it can be taken up by macrophages or taken up by other cells that will then initiate an additional response. So antibodies by themselves are very, very complex. They're not just there to bind. They have different functions attached to their back. So that's going to depend on the isotype. IgG. IgG. There are several ones: 1, 2, 3, 4. And so that's what the antibody does. It neutralizes the virus as it's outside of the cell and incapacitates it. A T cell is completely different. A T cell is interacting with cells. It doesn't interact directly with whatever you're trying to kill, bacteria, virus, whatever it is. So the T cell is going to interact with a cell that may be infected by a virus or a bacteria. And the way it does that is indirectly. So every single cell in your body, except for red blood cells, which is an exception, but carry molecules that are almost like a mirror of what's happening inside the cell. So inside the cell, obviously you have proteins being made, you have all the cellular machinery, you have the nucleus with the DNA in there. The cell is very, very complicated. But whatever is happening in the cell, there are bits and pieces of every protein that's inside the cell that is being shown on its surface. And the molecule that does that is called MHC or HLA in specifically for human leukocyte antigen, HLA. This is the molecule that you type for transplantation. So this is how we differ from each other. So you have six different ones, 6 class 1 and class 2. So there are different classes as well, depending on the peptide and depending on the T cell it interacts with. But the main point is here that the T cell interacts with a molecule on another cell, and that molecule presents bits and pieces of protein that come from the inside of the cell. So a cell that is infected by a virus will produce more viruses. That's what the virus does. It infects the cell, and then the cell is hijacked and produces more of the viruses. That's what the virus is using the cell for. And so the fact that the cell produces suddenly viral proteins, that will be shown on the surface. And so the T cell can recognize, okay, well, hang on, that piece of protein, that's not us. Us is our body. And if it's not us, T cell will recognize that as foreign. And then they will kill, if they're properly instructed beforehand, they will kill that cell. So these are two different ways to protect yourself against a viral infection. And of course, in a real life scenario where you're infected with an organism, you will have both simultaneously. It's not either or, you will have both. But in the case of a vaccine, to induce a particular response, you need to figure out what is your primary objective. Is it the B cell or is it the T cell? Because it will differ in what you manufacture. What is the vaccine? How is it made of? What is it made of? So do you want to have just those T cells floating around and capturing T cells that are infected and kill them? So you kill kind of the source of making more viruses, or do you just want to primarily neutralize whatever's floating around in terms of viruses? And so typically, if you go, for instance, for a B cell targeted or designed vaccine, what you will measure, sometimes exclusively, is you just take blood of a person, you isolate the plasma, and then you measure an antibody titer. A titer is nothing else than, it's a measure of concentration. How much of this antibody that's specific for virus do you have floating around in your blood? That's what a titer is. And so when you design a vaccine, or you try to figure out how effective it is, before you actually go into the real world and figure out whether people are actually protected from it. So if you first do preclinical studies on it, and you want to compare different vaccine designs to induce a B cell response, you will compare titers. So, that vaccine, that concentration of antibody is kind of 10 times higher than the other one. So that one's intrinsically chosen to be better just based on the antibody titer, if it's specific for the pathogen. So we're not measuring just total antibodies, we're measuring antibodies that are specific for the thing you want to protect against. And then, so you can measure a titer, and then you can also measure, it's a more functional way to assess how much protection do you get, is to do neutralization assays. So with live virus in the lab, you will see whether the antibody you produced, it's great if it binds the target, but it actually has to bind the target in a way that neutralizes its further replication. Because it can just bind, and then the virus doesn't give a shit. It just hooks up a couple of antibodies. Yeah, I'm just going to drag them around. It's fine. So an antibody has to somehow neutralize the virus. It doesn't just, if it just binds but doesn't neutralize it, it's not a good thing. It's not going to give you any protection. So with the T cells is always, and I'm a T cell immunologist, I mean, that's my domain of expertise. T cells are always more tedious to work with because you cannot just measure a titer with T cells. T cells, to measure whether you had an effective T cell response in humans, you can do it in animals. It's a bit more straightforward, but still not as simple as for B cells, but it's a bit more straightforward. But the issue is with what I told you, that you have these different proteins on the surface that display bits and pieces of the pathogen, and we all have different ones, right? I mean, identical twins have the same, but we all have different ones. So if you're talking about cytotoxic T cells, you're talking about MHC class 1. So you have 6 different ones. ABC, two A's, 2 B's, 2 C's. You got 1A, 1B, 1C from each of your parents. So that's why the compatibility usually for organ transplantation is higher within the family, because it's the blocking or the rejection mechanism goes via T cells. And so if you have a big mismatch of HLA, you will recognize another person's tissue just based on the fact that the things that are presented are completely different. So it doesn't even matter what's bound to it. Your T cells are gonna immediately realize, hang on, that's not even the same molecule. What the heck, and it's gonna immediately attack it. So that's a big difference between measuring B cell responses versus T cell responses. T cell responses have to almost be measured individually, and it's very, very tedious. It also means that because what's bound to those molecules, there are different ones. So you're thinking, well, they all present something, so what's the difference between them? So the specific amino acid sequence, so there's usually a tiny bit of protein, so protein is made of amino acids, so there's a sequence of roughly 8 amino acids for a class 1 molecule, so an 8 little links, 8 amino acids linked to each other is going to bind to it, and every molecule, so one of the ones you have, has a different affinity for the different sequences. So to make it very practical, so if you are infected by a virus, I'm infected by the genetically identical virus, I will present to my T cells a different part of the virus than you will. And to measure that, you need to do assays with blood and measuring, and so it's a complicated assay. It's not just taking plasma and doing an ELISA assay for antibody titers, which is very fast, very efficient. Here you're working with actually cells of the individual, and it's not even, it's highly likely that you will not be able to cover all of the possibilities because you don't know exactly. It's very complex and expensive to actually know every individual's HLA type. I mean, you can. I mean, that's what they do in transplantation, but it's actually quite costly. So if you're running a study, you want to know, okay, what did I induce in X, Y, and Z? It becomes very costly and very time-consuming to measure the T cell response in these individuals. And that's only going to be the blood. You don't even know what's happening in the tissue. So the point is that let me go back to, so how is a B and a T cell vaccine, how do they differ from each other? And so what are the components of a vaccine? I'm not going to go into details about, well, then there's excipients or things that are nasty that we don't want, blah, blah. Let's just talk about what you need to build a vaccine. Then I can't talk about manufacturing processes and what kind of residues might still be in there. But what you need is, what do you want to direct your immune response against? So a protein. And then something that constitutes a danger signal. The immune system is very complex and very powerful. The last thing you want is to give protein without a danger signal, because in that case, you're running the risk of inducing tolerance. So because the immune system is so powerful, it has a lot of checkpoints. Because the last thing you want is to launch an inflammatory response in your body uncontrolled, because then that means death very quickly. If you start to release inflammatory mediators from your immune cells all at once, That's a recipe for disaster. So everything needs to be controlled. Are we really infected? Is there really something bad floating around? Where I recognize something that's a bit different, but hang on, like, is it really bad? So there are different checkpoints, and there needs to be combination of signals, otherwise you're not launching an immune response. And that's to protect yourself against, well, everyone heard about autoimmunity. You know, suddenly if you have if your response that trickles, that is either directed against yourself or a response against a pathogen, but it's just too intense, you might just die. So that just is a testament to the power of the immune system. So giving just protein, even if it's a protein from a virus or from something foreign that should be attacked, if it's in the absence of a pure immunological danger signal, you could run the risk of inducing tolerance. That, for instance, is what people want to achieve in allergies, right? In allergies, you have this weird response against things that are innocuous, and there are specific regimens, for instance, with peanuts, where you give a little bit, without danger signal, and you try to tolerize the organisms against it. So the last thing you want is to tolerize yourself against something like a virus or a bacteria, because then you're like, your immune system is like, oh yeah, I'm cool with you. Yeah, you can come in. I'm cool with you. You don't want that at all. So the two main components of a vaccine are the target, protein, and a danger signal. And this danger signal is typically what we call the adjuvant. The adjuvant is something that's gonna annoy the immune system so that it's like, okay, what do you want me to react against? That's the antigen. And then the danger signal says, I really want you to fight that off. That's bad. It's not just, oh yeah, it's different. No, it's different, but it's also bad. So the typical reaction you get when you get a vaccine, intramuscularly, for instance, in the shoulder, like the pain and the swelling, all of that is wanted. You want to have an inflammation. You want to have that danger signal saying, hey guys, what's coming in right now, that's bad. If you wouldn't have any reaction, I mean, of course, like you can't always feel the reaction, but the reaction is there. It's the signal that's given. So the adjuvant is something that you want. It's not just there to, why do we have that? Why is it so painful? Because you want to give that danger signal. Otherwise, you're running the risk of tolerizing yourself against that. So there are various adjuvants. I'm not going to go through them. There's even, and of course, vaccination is a business, right? So of course, companies are going to try to find their own little adjuvant that does X, Y, and Z and patent it, and so that they have a trademark vaccine. Adjuvants is almost like a thing for itself. An adjuvant, and historically, I wouldn't always say that we understand fully what an adjuvant does. Some of them we know that we use them for decades, and suddenly we actually understand like how they actually work, because they were introduced empirically, 50 years ago, like, oh yeah, if we put that in, we get a good response and we protect people. But like no mechanistic clue of how we actually did that. And so that there is immunologically, like if you guys think that we understand all of it, like definitely not. Whoever thinks that, oh yeah, the immune system, I can explain you exactly how X, Y, and Z works, I wouldn't trust that person. There's so many still unknowns in the field of immunology and adjuvants. The problem becomes if people try to design something and they do their own research, and so if not everyone else has also looked at what it does, they just covered the basics and say, well, it does X, Y, and Z, and that's all the data we have. And the thing is, so adjuvants, because an immune response is so complicated, because you have various, just think about it, like just to give two extremes. So you have like what we call a Th1 versus a Th2 response. So Th1 is like classical response against a virus or bacteria. call it type 1 based on the cytokines that are being produced. And a type 2 response would be mechanistically fairly distinct, and that would be an allergic response against whatever, peanuts. The set of cytokines that are secreted are completely different. There's overlap, but we treat them quite distinctly, and then depending on how you categorize them, there's Th17, Th9. I mean, it depends how far down the rabbit hole you want to go, but depending on the signature of what you're inducing, that's how you basically almost can run a patent around, saying like, oh yeah, well, I found something, an extract from a plant somewhere in South America, and I realized, oh yeah, it has adjuvant activity, and it induces a set of cytokines or a combination of cytokines that so far has never been achieved with any other adjuvant. So I'm gonna try to use it and manufacture it and produce a vaccine with it, and I'm gonna be able to patent it because I have this kind of weird thing that no one expected. So the main thing is, if you look at it from above, yeah, it induces, it creates danger signals in terms of cytokines or cell signals. But everything does it slightly differently, you know, and knowing exactly what's good, what's bad, at the end of the day, on a population basis, you might also, if you test humans, you would test the adjuvant in humans. I would probably be comfortable saying that a lot of people would react differently. So that's why, I mean, a lot of things are tested in animals, in mice typically for immunology, because we have a lot of tools in immunology that run with mice. And of course, in mice, everything's homogenized, everything's standardized, because you will work in typically only in one mouse strain that are genetically identical. So of course, everything's going to be tight. Every animal is going to react exactly the same way. But then in humans, it might completely differ. And it's where also the field of immunology, I think, has learned a lot and is moving more towards actually doing more work in humans compared to mice, because a lot of things, cancer immunotherapies, other things, have never translated to human. Work beautifully in animals, and then suddenly you go to clinical trials and you realize it's all shit, doesn't work. And well, part of it has been also the massive shift with the microbiome, understanding how the microbiome impacts these things and reproducibility of results. But yeah, so the adjuvant is quite an important piece. And sometimes we don't fully understand what the adjuvant actually does. We can measure a few cytokines and say, well, it's immunogenic. But it might do other things that we don't measure. And that's really the point about vaccines, I think, and that's where one of the main issues is. The fact that if you design a B-cell vaccine, probably the only thing you're going to measure really are antibody titers. And then you're completely in the blind of what else it's doing. Are you going to measure the T-cell response? No, you're not. You might do a few, whatever, flow cytometry assays, whatever, cytokine, intracellular cytokine stains, blah, blah, blah. But you're not going to fully in detail understand what exactly did you do. You're just going to measure antibody titers. And I think potentially the blind spot is in all the things that we're not measuring that the vaccine potentially does. Because launching an immune response is never, with my understanding of immunology, you can never say that an immune response is entirely safe. Not on a large scale, because an immune response can always, once you understand the power of the immune system, launching it off is always associated with risk. And of course, being infected by something will carry the same risk. You will be infected by something, and then you might suddenly launch a response, and then suddenly you can't recover from the infection. And it has nothing to do with infection, because the pathogen might have already been cleared, but you launch something that somehow was not able to be terminated. And that's for instance what you also see in what we call long COVID, when you have the infection and then suddenly you're unable, your immune system is not going back to baseline. And again, immune system in its broader sense, the entire organism is not going back to baseline based on what the immune system did. And so coming back to the design of the vaccine, where so you want to have the antigen, you want to have the adjuvant. Well, to my understanding, that's basically was the lowest hanging fruit for instance, Russia and China. They went with traditional vaccines as fast as they could. Protein, adjuvant that has been used like in other vaccines beforehand. Let's go, let's do it. The annoying thing when working with protein, it's typically a recombinant protein, so protein that you overexpress in a bacterial culture, massive fermenters that produce that protein. It's tedious. You can't patent it because it's like protein and adjuvant. If you take an adjuvant that's already been out there, the protein you can't patent it. There's nothing to patent. So the vaccine itself is not patentable. But it's reasonably, like, you know what it should do, and it should be fairly effective, and you have a lot of data. And so that was not done for the mRNA. So the mRNA is a completely different technology. So instead of having the protein and the adjuvant, the reason why mRNA has gotten so much traction is it's a lot faster and cheaper to upscale. Protein, again, like it's heavy duty fermenters and then purification processes. It's tedious, it's boring, and it's not patentable. As soon as you're dealing with viral vectors, so not necessarily mRNA, but also viral vector vaccines, where it's like a piece of a virus that produces then the protein, the target, or mRNA, which encodes for the protein you want to target against. It's first of all, as soon as you're talking with genetic material, you can replicate and upscale it much faster. It's much more efficient. Much faster, and you can obviously, based on the technology, which hasn't been used so far a whole lot, you can patent manufacturing processes, or in the case of mRNA vaccines, also the membrane, the nanoparticle itself that will carry the mRNA, and also mRNA, the stability, how you stabilize it, all of that. There's new technology around that. So this is a completely different way to induce the response, where the host itself, you producing the protein. It's not what protein is given to you, but you produce the protein. The adjuvant, I think, when the vaccines were first given, the adjuvant wasn't super well established. So it's around the genetic material itself. So the adjuvant effect has actually, once it was rolled out, has been the subject of quite a few investigations to understand exactly how the adjuvant effect comes about. But yeah, so this is I think this is the basics. So you need to think about, vaccine is a very broad term. Its broad function is to induce an immune response that will protect you against something else, something that's already here, but more often than not something that you want to protect against that you haven't contracted yet. But it's very important to distinguish around those B cells, T cells. B cells easily measurable, but also like you don't measure it in depth. You don't sequence really the antibodies or something like that, but you measure a titer, and it gives you a good clue about the quantity of the material that will protect you against the target. T cell responses, and again, it's not mutually exclusive. You always have a bit of T cell, a bit of a B cell. With some of the properly designed T cell vaccines, if it's a short epitope, you might not get B cells at all, but that's an exception with very, very small peptide vaccines. But typically, you always have a bit of both. But it's really the T cell response where we all differ, and that could explain a lot of differences in efficacy, because because of the indirect way the T cell recognizes its antigen, and the fact that we're all genetically different. So I think I'll leave it at that as a little primer for vaccines. Let me know in the comments if you want to hear anything specific, if there are specific questions. If you want to have a bit more intro about the immune system, I don't really want to do it if there's no interest in it. The immune system is quite complicated, so I think it makes always sense to talk about it in the context of something that's interesting, either a disease or an application. There's a lot of hype around the immune system and cancer with different immunotherapies, checkpoint inhibitors, which have really, I think, revolutionized cancer immunotherapy or cancer therapy in general. Instead of relying on chemo and radiotherapy, there's now, depending on the cancer you have, you can have almost functional cure from cancer if you can launch the appropriate immune response. So yeah, that's me. Not sure what I'll talk about next. Maybe I'll get back to spirituality, which was the biggest incentive to launch this channel. But I'm happy to throw in some purely scientific posts in here if there's interest. Much love, everyone. See you next time.